Good day, everyone. Welcome again to another lecture of Hematology 2, MLS 417. This is Winbrent M. Corpus, your lecturer for today. This will be my last lecture for your batch. This will be about secondary hemostasis or coagulation cascade and fibrinolysis, which is the endpoint of the coagulation. The source of this discussion will be from your Products Hematology Clinical Principles and Application and some on Clinical Hematology by Staninger. So before discussing about the coagulation or the secondary hemostasis and other functions, so let's first discuss or define what is hemostasis. Basically, hemostasis is a complex physiologic process that keeps the circulating blood in a fluid state. Because in normal situation, the blood should be a fluid state, so there should be no clot inside the blood vessels. So when there is injury, um, it normally produces clot to stop the bleeding, which eventually confines the clot to the site of the injury and finally dissolves the clot as the wound heals. So when the hemostasis or the hemostasis system are out of balance, so for example, in cases of hemorrhage, or when there is uncontrolled bleeding, or when there is thrombosis, which is a pathologic clotting, this could lead to derangement that could eventually be life-threatening. So in hemostasis, there should be a balance between bleeding and clotting. So when there is excessive bleeding, it's dangerous. The same as with excessive clotting, like in um, thrombus formation, which eventually can go through the different circulation into your microvasculature, leading to embolism. Thus, decreasing the supply of nutrients and oxygen to the affected site. So there should be again balance in hemostasis between bleeding and clotting. So in the absence of single plasma procoagulant, may predispose the individual to lifelong anatomic hemorrhage, also to chronic inflammation and transfusion dependence. Conversely, the absence of a control protein allows coagulation to proceed unchecked and results in thrombosis, stroke, pulmonary embolism, deep vein thrombosis, and cardiovascular events. That is why I emphasize that in hemostasis, there should be a balance between clot formation and bleeding. So understanding the major systems, such as the involvement of our blood vessels, platelets, and plasma proteins are very important for us to know and for us to interpret laboratory results and to prevent, predict, and diagnose and manage hemostatic disease. So for example, when there is absence of plasma procoagulant, meaning they promote clotting, so the patient might be predisposed to again anatomic hemorrhage, inflammation, and transfusion dependence because they are they have no plasma procoagulant that promotes clotting, so they need to be transfused with a lacking coagulation factor. However, in the absence of control of protein that controls the coagulation, so there is excessive clot formation. So there would be thrombosis, as I've mentioned a while ago. In here, as you can see, there is deep vein thrombosis, wherein there is thrombus formation, more commonly in the lower extremities. And eventually, when the thrombus get dislodged from the site of the clot formation, it becomes an embolus. When the embolus, as you can see in the rightmost image, goes to the microcirculation, for example, in the brain, where there is lesser diameter of the capillaries supplying the cortex or any part of the brain, this could lead to embolic stroke, thus predisposing the person to a pathology. So thus leading to the demise of the patient. So again, let me emphasize that in hemostasis, there should be a control of bleeding and control of clotting. So there should be balance. I've mentioned the previous slide that in understanding hemostasis, we should understand the systems involved. So there is an involvement of the blood vessels, specifically your endothelium, Next will be the platelets, which produces some of the coagulation factors and the cofactors. And then we have the plasma proteins or the coagulation factors itself. So they are very essential in understanding and interpreting laboratory test results. For example, there is normal coagulation factor levels, but there is an abnormal platelet function. So this could lead to bleeding. The same as with your plasma proteins, when they are deficient, it could cause bleeding. So we use these three systems to prevent and predict and to diagnose and manage hemostatic diseases. So on the week 17, you will be learning about the disease of the secondary hemostasis and fibrinolysis. So in this one, as you can see in the left image, you can see platelets are involved in hemostasis, the same as your coagulation system, thrombin generation, and the blood vessels or the endothelial cells. So for the coagulation, the endpoint would be thrombus or clot formation. So there is coagulation, involvement of the platelets, and fibrinolysis. The same as with your fibrinolysis, there is decrease in coagulation since when we define as fibrinolysis, it lyses the fibrin. There would be decreased platelet function to avoid clot formation, and there would be increase in fibrinolysis, again to dissolve the blood clot that is formed. So, going to the rightmost part of the slide, the right image, these are the major components again of the hemostasis. What are those? These are the endothelium, the platelets, and the plasma proteins. As you can see here, when there is vascular injury, the first response is coming from the endothelium, so or the blood vessels. So they vasoconstrict to decrease the pressure or the blood flow. Next would be there would be platelet activation because when there is an exposed tissue factor or collagen, the platelet will be activated and will eventually activate also the coagulation cascade and vice versa. So for the platelet activation, the endpoint would be platelet plug formation. For the coagulation cascade, this would eventually activate the extrinsic and the intrinsic, and together they will activate the the endpoint pathway, which is the thrombin formation, and eventually converting the fibrinogen to a stable fibrin clot. So later on, we'll be tackling about the coagulation cascade and the coagulation factors. And the endpoint of this system would be blood clot formation. So this coagulation cascade comprises the secondary hemostasis. And when the blood clot had served its purpose, so the plasmin, which is involved in the fibrinolysis, will be activated, and eventually we will be having the fibrinolysis or the clot dissolution and clot degradation. So just remember the three major components of the hemostasis. We have the blood vessel or the endothelium, we have the platelets, and the plasma proteins, which are the coagulation factors. So let me emphasize again the system involved in hemostasis. We have the blood vessels or the endothelium and the platelets, which are involved, both are involved in a primary hemostasis, and the plasma proteins or the coagulation factors are function or the purpose of which is for the secondary hemostasis. So again, let me define hemostasis. This involves interaction of vasoconstriction, as mentioned a while ago, platelet adhesion and aggregation, which are the function of the platelets, and coagulation enzyme activation to stop bleeding via clot formation. So the coagulation system, similar to other humoral amplification mechanism, is complex because it involves the blood vessels, platelets, plasma proteins, that are very important um, in disease states such as bleeding or hemorrhage or thrombus formation. So these are the key elements or the key systems that is involved in hemostasis. So for the primary hemostasis, this refers to the role of blood vessels and platelets. Remember that when we say primary hemostasis, it is the function of the blood vessels and the platelets in the initial response to a vascular injury. As mentioned a while ago in the previous slide, that when there is injury to the blood vessels, the first response would be vasoconstriction and platelet plug formation. And then when we say secondary hemostasis, it is the activation of series of coagulation proteins or factors seen in the plasma, which are mostly serine proteases, so they are enzymes, to form a stable fibrin clot. So these proteins circulate as inactive zymogens or proenzymes that will eventually become activated during the process of coagulation. So the endpoint of which is to generate thrombin and to convert the fibrinogen to a localized fibrin clot. So the hemostasis doesn't end with the primary and secondary hemostasis because the final event of the hemostasis is always fibrinolysis. We cannot afford na magstay lang yung clot sa ating blood vessels because normally there should be no clot because I've mentioned a while ago that in vivo, the blood should be in fluid state. So in fibrinolysis, there would be gradual degradation or digestion and removal of the fibrin clot as the healing occurs. So again, primary hemostasis is the function of your blood vessel or endothelium and platelets. Secondary hemostasis involves your coagulation factors. And eventually, the last or the final event of hemostasis is fibrinolysis. So as shown in table 35.1, these are the processes that is involved in primary hemostasis and secondary hemostasis. Please do familiarize them as they would come out in your examination. I've mentioned a while ago that about the intrinsic and extrinsic and the common pathway. So I expect you all to memorize and familiarize the pathway since this is very important in understanding the deficiencies or the pathologies that will be discussed on week 17. So in intrinsic factor, it is this usually starts when there is surface contact with, for example, with the collagen, and for the extrinsic pathway, usually via tissue damage. So the intrinsic and the extrinsic pathway will meet in the factor 10 in a common pathway and eventually will lead to the formation of the stable fibrin clot by the action of your factor 13A. So just familiarize the steps and also the coagulation factors that is involved in intrinsic, extrinsic, and common pathway. Another one to remember is that when we measure intrinsic pathway, we test for APTT because APTITI, it's for the intrinsic pathway. For the extrinsic pathway, we measure for PT or the prothrombin time. So PET, PET for extrinsic pathway. So remember that. So we also have just an introduction to the diseases of the secondary hemostasis. We have coagulation disorders. It could either be in a form of excessive or inadequate clotting or a deficiency of clotting factors. So we have hemophilia A, hemophilia B, and C. We also have von Willebrand disease and vitamin K deficiency. So this I will not be elaborating on this because this will be discussed on week 17. But remember, the endpoint is the formation of the stable fibrin clot. So these table 35.6 are very important in understanding the coagulation system because we have here the list of the coagulation factors and also take note of the names or the other names and their function. So as you can see here, factor number 1 is called the fibrinogen. And next would be the factor 2 or the prothrombin. That factor 3 is the tissue factor. Factor 4 is the ionic calcium. And then we have a cofactor 5. And then we have 7. We have 8, which is the anti-hemophilic factor, which is deficient in hemophilia A. We also have von Willebrand factor, which is associated with your factor 8. We'll be discussing that later. So von Willebrand factor acts as a shield of the factor 8. We have factor 9, which is the Christmas factor, which is deficient on hemophilia B. We have factor 10 or the Stewart-Prower factor, which is deficient on your hemophilia C. We have factor 11, 12, which is the Hedgeman factor. And then we have prekallikrein or the Fletcher factor, the pre-K. Remember, this might come out on your board exam because in our time, factor 12 was asked, the Hedgeman factor. We also have the high molecular weight kininogen or the HMWK, which is also called the Fitzgerald factor. Factor 13 is the FSF or the fibrin stabilizing factor. So take note for their other names because this might be asked on your exam. So we have plasma transports. Uh, the plasma transports at least 16 procoagulants. They are also called again the coagulation factors. So they are nearly all glycoproteins synthesized in the liver, although some are synthesized or produced in the monocytes, epithelial cells, and megakaryocytes. So 8 of which are serine proteases or inactive form in zymogens. As you can see here, these are factor 2, factor 7, factor 9, 10, 11, 12, prekallikrein. Yes, prekallikrein. And also we have transglutaminase factor 13. So take note of that. And others would be a cofactor. So when a procoagulant becomes activated, so a lowercase, yung letter A will appear behind the numeral. For example, activated factor 7, so this would be termed as 7A. So both zymogens and cofactors become activated in the coagulation process. So take note of these following other names and the numerical designation of the coagulation factors. So we can categorize the plasma components of the coagulation system into three. We have first the zymogens, which are the prekallikrein, factor 12, 11, 9, 10, 7, prothrombin, and factor 13. We also have the cofactors, which is required for the activation of your zymogens. We have the high molecular weight kininogen, the tissue factor, factor 8, 5, protein Z and S, and thrombomodulin. And then in your coagulation cascade, there should be a control proteins because we cannot afford excessive clot formation. So we have antagonist of the zymogens. We have the antithrombin, which is the major control protein for the coagulation cascade. We have the heparin cofactor 2, the TFPI or the tissue factor pathway inhibitor, which basically inhibits the tissue factor in the extrinsic pathway. We have protein C, we have alpha-2 macroglobulin, alpha-1 antitrypsin, and ZPI or the protein Z-dependent protease inhibitor. So as you can see here, the zymogens are the fibrinogen substrate. So they are required for the fibrin formation or the clot formation for the clot to be stable. As you can see in the figure 35.4, this is the simplified coagulation pathway, wherein it could start in the tissue factor 7 pathway or the extrinsic, or it could start in the factor 12 prekallikrein high molecular weight activating the factor 11 in the intrinsic pathway and eventually meeting at the common pathway at factor 10. So the goal of the secondary hemostasis is basically to generate the key thrombin enzymes and produce the fibrin, which is the localized thrombus. Again, I would really emphasize it should be localized thrombus formation. So kung saan yung site of injury, dapat doon lang tayo mag-form ng clot or stable clot. So these are some key points that should be remembered. Serine proteases that are coagulation factors that are considered as serine proteases are factor 2, 7, 9 to 12, and the prekallikrein. And the transglutaminase is only factor 13. So, di ba, I've mentioned a while ago that when your zymogen or your inactive zymogens are activated, so there would be a subscript that will be added. For example, in your factor 7, it will become factor 7A with a cofactor of tissue factor. And then their substrate would be factor 9 and 10. So be familiar with this table 35.7. And then for the other plasma procoagulants, we have box 35.2, we have fibrinogen, factor 13, phospholipid, calcium, and 1 milligram factor. So these cofactors are essential in the formation of clot formation. So without these um, cofactors, the coagulation cascade will be delayed, or it could be displayed as prolonged PT or prolonged APTT laboratory test results. So the coagulation factors involved in the secondary hemostasis could be divided into several categories. So first is the vitamin K dependent prothrombin group. So this involves the prothrombin or the factor 2, the factor 7, factor 9, and factor 10. And regulatory proteins such as your protein C, S, and protein Z, which are dependent on vitamin K, thus their name vitamin K dependent prothrombin group, during synthesis to produce a functional structure. So they are named the prothrombin group because their structural resemblance to the prothrombin. So all of the 7 proteins have 10 to 12 glutamic acid units near their amino acid terminal. All except proteins S and protein Z are serine proteases when activated. So the protein S and Z are cofactors. Again, protein S and Z are cofactors. So vitamin K is essential again in this group. So vitamin K is a quinone found on green leafy vegetables. So when there is a case that the patient is not having or not consuming food rich in vitamin K, so for example, what are the coagulation factors that could be affected or most likely be affected? So you choose between prothrombin, factor 7, 9, and 10. So they are also produced by intestinal organisms such as the bacteria Bacteroides fragilis and Escherichia coli. So the vitamin K catalyzes an essential post-translational modification of the prothrombin group proteins. So take note of that. That is why vitamin K is very important in this group. So on box 35.3, here are the food sources high in vitamin K. So if you will be given a case, so for example, meron daw siyang vitamin K deficiency, so which of the following could be added on the diet? So these are examples. So syempre, mahirap pag-memorize ng mga coagulation factors. So we have a mnemonic for vitamin K dependent clotting factors. So we have 2 plus 7 is 9, not 10. So 2, 7, 9, 10. So others, in our time, meron kami 9, 10, 72. So you can choose kahit saan sa kanilang dalawa. So again, mapansin nyo if you have siblings, mga newborn, so they are given 0.5 ml IM vitamin K because babies or the newborn infants, mga bago lang anak, so they lack bacteria on their intestines if wala pa sila naka-breastfeed. So for us to prevent yung mga bleeding in or hemorrhage in the newborn, so they will be given vitamin K injection because as we know, factors 2, 7, 9, and 10 are vitamin K dependent. So without the vitamin K coming from the activity of your Bacteroides fragilis and your Escherichia coli in the intestine and wala pa yung diet ng baby, hindi pa sila makakain ng green leafy vegetable, kaya yun yung rationale bakit sila bigyan ng vitamin K para hindi maapektuhan yung clotting factors 2, 7, 9, and 10. So pag magtanong yung mga kamag-anak nyo, what is the importance of giving vitamin K injection in newborn? So kasi po, we have vitamin K dependent clotting factors which requires, kaya nga vitamin K, they require vitamin K for it to be functional, yung factor 2, 7, 9, and 10. Let me explain the significance of vitamin K in the coagulation factor 2, 7, 9, and 10. So I said earlier that vitamin K catalyzes an essential post-translational modification of the prothrombin group proteins. So we have the G-carboxylation of amino acid amine terminal glutamic acid shown in figure 35.5, wherein the glutamic acid portion, as you can see in the non-active coagulation factor, is modified to a G-carboxyglutamic acid when a secondary carboxyl group is added to the G-carbon. So with the two ionized carboxyl group, the G-carboxyglutamic acid gain a net negative charge. So this net negative charge enables them to bind to ionic calcium. So di ba, calcium is very important as a cofactor. The bound calcium enables these vitamin K dependent coagulation factors to bind negatively charged phospholipids to form complexes with other coagulation factors. So without the vitamin K, this process would not proceed, so there would be non-active coagulation factors in 2, 7, 9, and 10. Yeah. So without also the calcium, the process will not proceed. So in cases of your vitamin K deficiencies or in the presence of your anticoagulant drug, for example, Coumadin, which is a therapeutic inhibitor of vitamin K, the vitamin K dependent coagulation factors here, 2, 7, 9, and 10, are released from the liver without the second carboxyl group added to the G carbon. So, di ba dapat they will form net negative charge for it to bind calcium. So, these called DES G carboxyl proteins or proteins induced by vitamin K antagonist factors will lead to lack of second carboxyl group. So again, they cannot bind to calcium and phospholipids. So they cannot participate in the coagulation factor or the coagulation reaction or system. So that's the importance of your vitamin K. So ang target protein niya is your glutamic acid. Dapat mag-add siya ng second carboxyl group for it to gain a net negative charge for it to attach to calcium, or the ionic calcium specifically. So for the antagonist of your vitamin K, we have this warfarin and coumadin. So this would be the mechanism of the vitamin K antagonism for your oral anticoagulant warfarin and coumadin. So warfarin blocks this step. As you can see in the rightmost image, yung reductase, yun yung ginablock niya, yung vitamin K oxide to vitamin K. Yan yung target for your anticoagulant warfarin and coumadin. So next to your vitamin K dependent procoagulant or the coagulation factors, we have the cofactors in hemostasis. So we have here in table 35.10, we have procoagulant cofactors such as tissue factor, the factor 5, the factor 8, and the high molecular weight kininogen. So these are transmembrane receptors for factor 7A, or we have here the tissue factor, and they are found on the extravascular cells surrounding the fibroblast and smooth muscles. So when these cofactors bind with its particular serine proteases or your zymogens, when bound to their cofactors, these serine proteases gain stability and will have an increased reactivity to the coagulation system. So under normal conditions, so the tissue factor is not expressed on the blood vessels. So when there is vessel injury or breach to the endothelium, these will expose the blood to the subendothelial tissue factor-bearing cells and will eventually lead to the activation of coagulation through the factor 7A in cases of vessel injury. So thus the activation of factor 7 is rate limited by only injury itself, so breach to the endothelium. So tissue factor is expressed especially in high levels in the brain, lungs, placenta, heart, kidneys, and testes. So in cases of inflammatory conditions and sepsis, leukocytes and other cells can also express tissue factor and could eventually initiate the coagulation cascade via the extrinsic, since the tissue factor belongs to the extrinsic pathway of the coagulation cascade. So remember this table, the cofactors and their functions, and where they bind to. Next, we have factor 5, which is a glycoprotein circulating in plasma and also seen in platelet alpha granules. So this is a cofactor to factor 10 or the prothrombinase complex. As you can see here in the table, factor 5 is a procoagulant cofactor which binds to 10A. We also have factor 8 which is a cofactor to factor 9. So factor 9A yung kaniyang ginabind. Next, we have the high molecular weight kininogen, which is a cofactor of factor 12A and prekallikrein in the intrinsic contact factor complex. This activates coagulation in conditions where there is foreign objects such as mechanical heart valves, bacterial membranes, or high levels of inflammation. So, high molecular weight kininogen promotes coagulation via the contact factor system. Di ba, kasali dito yung factor 12 and prekallikrein in the intrinsic pathway. So in the exam, when you read about there is presence of mechanical heart valves in the patient, so think of high molecular weight kininogen and the intrinsic factor cofactors and procoagulants. We also have other cofactors such as your thrombomodulin, which functions as a control in your protein C and serves as antifibrinolytic in your thrombin activatable fibrinolysis inhibitor. So they both, or the thrombomodulin binds to the thrombin. So thrombomodulin is a transmembrane protein which is constitutively expressed by the vascular epithelial cells and is a thrombin cofactor in the protein C pathway. So together with the thrombomodulin and thrombin, they activate protein C, a coagulation regulatory protein. And once the thrombin is bound to your thrombomodulin, so thrombomodulin plus your thrombin, it loses up its procoagulant ability or to form clot to activate factor 5 and 8. And through activation of the protein C, this leads to the destruction of your factor 5 and 8, thus suppressing further generation of thrombin. So wala tayong clot formation doon. Thus, it is considered as a negative feedback loop. But when your thrombin is bound to your thrombomodulin, it could also activate your thrombin activatable fibrinolysis inhibitor at a fibrinolysis inhibitor. So, dalawa yung pwedeng action ng thrombomodulin. Pag yung thrombomodulin, nag-act siya sa protein C, it acts as a control. When the thrombomodulin binds naman sa thrombin, it could also be antifibrinolytic via your TAFI. We also have protein S and Z, wherein your protein S is a cofactor of your protein C and TFPI, or your tissue factor pathway inhibitor, and protein Z, a cofactor of your Z-dependent protease inhibitor, or your ZPI. Just remember the two. So they are not commonly asked in the exams, but just remember them. Next, we have factor 8 and your VWF, or your von Willebrand factor, which is very important in the coagulation. So factor 8 and von Willebrand factor are the key proteins for the hemostasis. So for factor 8, these are produced, or it is produced in the hepatocytes in the liver, microvascular epithelial cells, most commonly in the lungs. So they are involved in all protective mechanisms to avoid blood loss. So when the free factor 8 is when there is free factor 8 in the plasma, so they become unstable. So that is why your factor 8 always circulates with your von Willebrand factor. So yung von Willebrand factor here serves as a shield or defense for the factor 8, for factor 8 not to be unstable. So factor 8 is inactivated by your protein C, and then your factor 8 and factor 9 are governed by your X chromosome. So I mentioned a while ago about hemophilia A and B, wherein your hemophilia A is when there is deficiency of your factor 8, and the hemophilia B when there is deficiency of your factor 9. So as you can see here, the von Willebrand factor, factor 8 complex, so your von Willebrand factor attaches to your factor 8 so that it provides three other active receptor sites. So yung von Willebrand factor nyo, meron siyang platelet membrane binding site for your GP1B, 9.5 and GP2B3A, and also for your collagen. I've mentioned that your von Willebrand factor is a shield or acts as a shield or defense for your factor 8. So when your factor 8 is not bound to your von Willebrand factor, they deteriorate more rapidly. So dapat merong one brand factor na attached sa kanya. And then your factor 8 deteriorates more rapidly than the other coagulation factors in the stored blood. So when there is a question that which of the following coagulation factors will most likely be absent in stored blood, it should be factor 8. So in third component plasma, the von Brand, or I mean the factor 8 level drops to 50% after 5 days. So remember this. So pag na-store na yung plasma, factor 8 levels will decrease 50% after 5 days. Now, let's go to your von Willebrand factor, or your VWF, which is a large multimeric glycoprotein that participate in platelet adhesion and transport factor 8. Again, it transports factor 8. So the epithelial cells release ultra-large multimers of von Willebrand factor in the plasma. They are usually 240,000 Daltons each. So these are produced by epithelial cells and megakaryocytes, and they are also stored in the alpha granules in platelets in the Weibel palade bodies. Again, take note of this. This was asked in your board exam. Von Willebrand factor are stored in the Weibel palade bodies in the epithelial cells. And then they are destroyed by Adams 13 in the blood vessels with high shear stress. By the way, Adams 13 stands for a disintegrin and metalloprotease with a thrombospondin type 1 motif member 13. So when you are asked what's the meaning of Adams 13, you know. Again, your von Willebrand factor are released in ultra-large multimers. So they are destroyed by Adams 13 in the blood vessels with high shear stress. I've mentioned the previous slide that your von Willebrand factor together with your factor 8 serves as a receptor or additional receptor. So von Willebrand factor is or has 4 additional receptors. Primary platelet surface receptor of von Willebrand factor is GP1B95, and we have here the arginine glycine aspartic acid or the RGD sequences found on your von Willebrand factor also bind a second platelet receptor, GP2B3A, which is involved in the platelet aggregation. So, dalawa yung platelet receptor ng von Willebrand factor, your GP1B95 and your GP2B3A. So, these receptors also are involved sa mga diseases that will be discussed later on your or in your primary or your secondary hemostasis disorders. So your VWF also binds to collagen. As you can see there, there is 4 binding sites. And then it also binds to your factor 8 because diba, it acts as just the shield or the transporter of your factor 8. So when there is deficiency of your von Willebrand disease, or I mean von Willebrand factor, there would be severe bleeding and symptoms associated with abnormalities in von Willebrand factor. So we have here von Willebrand disease, an inherited disorders, which usually is categorized by type 1, type 2, and type 3 with the following defects. Please familiarize this table. And they could be autosomal dominant or recessive in nature. So von Willebrand disease is also associated with factor 8 deficiency since, di ba, when your factor 8 is freely floating or moving just as factor 8, they are unstable. So madali lang sila ma-destroy. So when there is von Willebrand disease, this would be linked to factor 8 deficiency. Because, again, your von Willebrand is the chaperone of your factor 8. So they say that the levels vary by ABO blood type. So researchers found out that group O have lower levels of von Willebrand factor than any blood group. So this is just a fun fact about von Willebrand factor. Just remember, von Willebrand disease is an inherited disorder. It could either be autosomal dominant or recessive. and associated with factor 8 deficiency since these transports factor 8. Next, we have factor 11 and the contact factors. So these are called contact factors because they are activated by contact with negatively charged foreign surfaces. So such as yung prosthetic heart valves, bacterial membranes, and etc. So the contact factors includes factor 12, high molecular weight kininogen or the Fitzgerald, and the prekallikrein or the Fletcher. So the contact factor complexes includes the high molecular weight kininogen prekallikrein factor 12A, which eventually will activate factor 11. So the factor 11A is an activator of the factor 9. So as you can see in the rightmost image, so when your prekallikrein attached to your high molecular weight kininogen, this will cleave or this will be cleaved by your 12A to kallikrein, and then your kallikrein will um, cleave your 12 into 12A, thus activating your 11 to 11A, and eventually your 9 will be activated to your factor 9A. And then your factor 9A together with your 8A plus the calcium will activate the common pathway. So again, factor 12A transforms the prekallikrein, a glycoprotein that circulates bound to your high molecular weight kininogen, into its active form kallikrein, which cleaves the high molecular weight or HMWK to form the bradykinin. So the factor 12 and the prekallikrein are zymogens that are activated to become serine proteases. But when we compare it to your high molecular weight, non-enzymatic cofactor siya. So again, yung zymogens dito na part is your factor 12 and your prekallikrein, and yung non-enzymatic cofactor is your high molecular weight kininogen. So the endpoint would be the activation of your common pathway. Again, bakit sila tinawag na contact factors? Because they are activated by contact with negatively charged foreign surfaces such as your mechanical heart valves and bacterial membranes. So what makes your factor 11 and your contact factor deficiencies, for example, deficiency of factor 12, high molecular weight kininogen or prekallikrein, is that they do not cause clinical bleeding disorders unless they are so severe that the deficiencies prolong the laboratory tests such as your PT and APTT, thus needing investigation. So again, for your mnemonics, 12 and high molecular weight prekallikrein does not have or does not cause clinical bleeding disorders. So take note of this one. So to continue, factor 12 is activated in vitro, again outside the body, by negatively charged surfaces such as non-siliconized glass, kaolin, or ellagic acid, which are used as test reagents for PT and APTT. And then in vivo, inside the body, they are activated by foreign materials such as your stents, valve prosthesis, and bacterial cell membrane. Your factor 11, activated by the contact factor complex and also by thrombin during coagulation cascade generated from tissue factor activation. So the deficiencies of factor 11, or what we call as the Rosenthal syndrome, only produces mild and variable bleeding. So just a mnemonic, so 2 roses, 1 factor 11 deficiency. So take note of the Rosenthal syndrome for factor 11 deficiency. So next would be the thrombin, which is the main enzyme since it is a protease, serine protease of the coagulation pathway. So the primary function of the thrombin is to cleave the fibrinopeptide or FPA and B from the alpha and beta chains of the fibrinogen molecule, thus activating the spontaneous fibrin polymerization and the beginning of fibrin clot formation. So this also increases coagulation mechanism by activating factor 5 and factor 8 and factor 11 to produce more thrombin. So this also activates factor 13, which serves to cross-link and stabilize the fibrin clot. Your thrombin also initiates platelet aggregation. And then again, I've mentioned here a while ago that when thrombin binds to your thrombomodulin, this will activate protein C to suppress coagulation, and also this could activate the TAFI to suppress fibrinolysis. So that's the function of your thrombin. So there are many functions. There are many functions of your thrombin. Meron siyang multiple autocatalytic functions. So thrombin is a enzyme ha. It is a key protease of the coagulation factor or pathway. Next, we proceed to fibrinogen structure and fibrin formation in factor 13. So for the fibrinogen, this is the primary substrate of thrombin as discussed a while ago, which are converted to soluble fibrinogen to insoluble fibrin clot. So this is involved also in platelet aggregation wherein there is a link activated platelets via GP2B3A platelet fibrinogen receptor. So also fibrinogen is synthesized in the liver. Normal values would be 200 to 400 milligram per deciliter. And also since it is synthesized in the liver, so diba, fibrinogen is considered as an acute phase reactant or your APR. So we expect that the levels of fibrinogen increases in cases of inflammation and infection. So they're also seen or produced in the platelet alpha granules, wherein they are absorbed, transported, and they are released when needed. So as you can see here, fibrinogen is involved in the platelet interaction or aggregation. So the fibrinogen attaches the two receptors, the GP2B3A, seen in the platelet membrane. So that's the importance of the fibrinogen rather. So this is involved in a platelet aggregation. Next, we have factor 13. So factor 13 is considered as heterodimer because this has two subunits. First subunit is the alpha subunit, mostly produced by the megakaryocyte and monocytes. And the beta subunit is produced in the liver. Another name for your factor 13 is the fibrin stabilizing factor. Why is it called such? Because as you can see in the image, so when your factor 13 is activated by your thrombin in the presence of calcium, it will become factor 13A. So this factor 13A will catalyze the cross-linking of fibrin polymer to a cross-linked fibrin, which is considered as the stable fibrin clot. So as you can see here, in the presence of ionized calcium, this produces a stabilized fibrin clot, which will be eventually removed by the fibrinolysis, which is the endpoint of the hemostasis. Now let's summarize the three main group for your coagulation factors. We have the fibrinogen group, we have the 1, 5, 8, and 13. These are not vitamin K dependent and they are consumed during coagulation. So we expect that they are absent in the serum because they are consumed during coagulation. So in serum, we have clotted blood in there, but they are present in the adsorbed plasma. Next, we have the prothrombin or the vitamin K dependent coagulation factors. We have the 2, 7, 9, and 10 or the 9, 10, 72 or 72, which are present in the serum except the factor 2 or the thrombin. And then they are absent in adsorbed plasma. So next, we have the contact factors. We have factor 12, 11, prekallikrein, and high molecular weight kininogen, which is also similar to your fibrinogen, which are not vitamin K dependent. But they are present in the serum as compared to your fibrinogen that is absent in the serum. So they are also similar to your fibrinogen group that is present in adsorbed plasma. So take note of this table since this is very useful in your mixing studies that will be discussed on your laboratory. And just a recap for the other names of your coagulation factors from factor 1 to 13. So we have a mnemonic from Pakistan to China, people pack as communist style producing havoc freely. So personally, I do not use this mnemonic because it's easier to memorize them one by one. So for factor 1, it is fibrinogen. Factor 2 is prothrombin. Factor 3 is the thromboplastin or the tissue factor. Factor 4 is your ionized calcium. Factor 5 is your proaxelerin. Factor 6, take note, this has no name or no designation. Your factor 7, your proconvertin. Don't confuse with factor 5, which is the proaxelerin. Factor 8 is the anti-hemophilic factor, which is deficient in hemophilia A. Then factor 9 is the Christmas factor, which is deficient in hemophilia B. And then factor 10 or the Stewart-Brower factor. And we have factor 11 or the PTA or your plasma thromboplastin antecedent. We have factor 12 or the Hedgeman factor, the glass factor, which is considered diba, under the contact factors. And we also have the factor 13 or the fibrin stabilizing factor, as you seen on the previous slide, that factor 13A stabilizes or cross-links the unstable fibrin clot to a stable fibrin clot. So remember this slide. This is very important. Remember, I've mentioned that we have different pathways in the coagulation cascade. We have the intrinsic pathway, we have the extrinsic pathway, and the common pathway. So let's first briefly discuss the intrinsic pathway. So this is considered, or the intrinsic pathway is considered to be the dominant type of pathway that is involved in the coagulation in vivo. So intrinsic because all the components in the system are found in the coagulation, or I mean in the circulating blood. So first, the contact phase begins with factor 12 absorption to the negatively charged surface of the vascular collagen or prosthetic heart valves and etc. So the prekallikrein circulates as a complex with a cofactor, high molecular weight kininogen. So di ba pair sila, your prekallikrein plus your high molecular weight kininogen. So this complex is absorbed in vivo to the negatively charged surface with factor 12. And then your factor 11 also complexes with your high molecular weight kininogen. So once the contact group is assembled, your factor 12 undergoes a conformational change in the presence of the kallikrein, which accelerates, as you can see there, the conversion rate with enhancement by the high molecular weight kininogen. So when there is formation of your factor 12A with the presence of your high molecular weight kininogen, this will activate the factor 11 to your factor 11A. So your factor 11A in the presence of your calcium, because diba, factor 11 is a enzyme, so it will cleave the substrate factor 9 to form the factor 9A. This reaction requires calcium. Take note, your change of your factor 9 to your factor 9A involves calcium. And then your factor 9A in the presence of your factor 8A will be involved in the activation of your common pathway. So I've mentioned that your factor 9A in the presence of your 8A, calcium and phospholipid, will activate the common pathway. So here is the common pathway highlighted in orange. So the activation of the factor 10 seen in the common pathway begins when there is presence of 9A, 8A, calcium, and phospholipid. So this will eventually form your 10A, and then will activate your factor 2 to factor 2A, and your factor 2A has two pathways. It will either activate your factor 13 to form your factor 13A, or it will activate your fibrinogen to form the fibrin. And diba, we know that your factor 13A is a fibrin stabilizing factor which complexes or cross-links your fibrin to form the cross-link fibrin or the stable fibrin clot. So that is your common pathway. In addition to intrinsic pathway, we also have extrinsic pathway, which is much less complex than the intrinsic pathway. So basically, the extrinsic pathway begins with the tissue factor, or your 7, or coagulation factor 7, or the tissue factor, which is a lipoprotein released from cell membranes into the plasma when there is, again, vascular endothelial injury. So extrinsic activation of the common pathway occurs when there is factor 7 and calcium formation to activate the factor 10 to 10A and the rest would follow the same as your intrinsic pathway. Kaya nga tinawag na common pathway because the intrinsic and the extrinsic pathway share this pathway to form the cross-linked fibrin. But the main difference of the intrinsic and the extrinsic pathway is that in extrinsic pathway, this requires breach to the vascular endothelium, while in your intrinsic pathway, this requires exposure to contact factors. This is just the summary of the intrinsic, extrinsic, and the common pathway of the coagulation cascade. Just take note of the coagulation factors involved in each of the pathway and surely you will not be lost. So again, the endpoint of the extrinsic, intrinsic, and the common pathway is the formation of the stable fibrin clot. So in normal state, we cannot afford to have excessive activation of the coagulation cascade or system. Thus, we have these coagulation regulatory mechanisms. So listed in table 35.11 are the coagulation regulatory proteins involved in controlling or limiting the activity of the serine proteases and the cofactors. So we have tissue factor pathway inhibitor, thrombomodulin, protein C and S, antithrombin, heparin cofactor 2, protein Z-dependent protease inhibitor, protein C inhibitor, your alpha-1 protease inhibitor, also called your alpha-1 antitrypsin, and lastly, your alpha-macroglobulin. So these regulatory proteins are involved obviously, in regulating the coagulation system. First, we have the antithrombin 3 or the AT3 is a member of a serpine superfamily. What's the meaning of serpine? It's the serine protease inhibitors, which are usually or mostly produced in the liver. We also have alpha-1 antitrypsin, which is a inhibitor of thrombin, factor 11A and kallikrein. We have C1 inhibitor, which inhibits factor 12A, 11A, and kallikrein. And then alpha-2 macroglobulin, which inhibits thrombin and kallikrein. And unique for alpha-2 or another thing for alpha-2 macroglobulin is that the rate of thrombin inhibition is lower than antithrombin 3. And also it inhibits the plasmin. Next will be the heparin cofactor 2, which is considered as the minor inhibitor of thrombin. As you can see, majority of them inactivates or inhibits the thrombin. And then your antithrombin is considered as the physical physiologic inhibitor of the thrombin and factor 10A. As you can see in the figure 35.12, your antithrombin inhibits factor 11A, 9A, 10A, and your thrombin. So it also inhibits factor 12A, 11A, and 9A. So take note of that. We also have protein C and S, which are considered as a natural inhibitor of the coagulation. They are synthesized. Their synthesis rather is dependent on vitamin K, as we mentioned in your vitamin K dependent procoagulants and cofactors. And then they're activated by thrombin in the presence of the thrombomodulin. Diba kanina when thrombin and thrombomodulin attaches to each other, as you can see in 35.14, this activates the protein C and S, and then cleaves factor 8 and 5. So as you can see there, the PC or the protein C will become activated protein C and will inactivate the 5A and the 8A. So protein C is inactivated by your protein C inhibitor, obviously. So we also have inhibitors of the tissue factor pathway, or also called as the tissue factor pathway inhibitor, or the TFPI. So this is the principal regulator of the tissue factor pathway. As you can see there, your tissue factor pathway inhibitor inhibits the 10A. So the TFPI binds the complex of tissue factor 7A, 10A in a 10A dependent mechanism. So for step 1, the TFPI first bind to the factor 10 and inactivates it. So in the step 2, the TFPI or the tissue factor pathway inhibitor 10A complex binds and inactivates the cell-bound tissue factor 7A complex, preventing the activation of the 10A. So that's the mechanism of the tissue factor pathway inhibitor. But basically, please, ito lang yung kailangan yung matandaan dito. So, tissue factor pathway inhibitor inhibits the tissue factor pathway. Yun lang. We are now in the end of the discussion. So we are in the fibrinolysis. So this is considered as the final stage of the hemostatic activation. Again, this is the final stage of the coagulation. So fibrinolysis is defined as the systematic accelerating hydrolysis of the fibrin or the clot by plasmin. So hours after the thrombus formation or the clot formation, in response to inflammation and coagulation, this will activate the fibrinolytic process. This will release fibrinolytic proteins such as your TPA or tissue plasminogen activator and UPA or the urokinase plasminogen activator. So these proteins convert your fibrin-bound plasminogen into the principal enzyme of the fibrinolytic system, as you can see in the image, from the plasminogen to your plasmin. So this plasmin is the key enzyme in the fibrinolysis. So the fibrinolytic process degrades your fibrin, which aids in restoring normal blood flow during vascular repair. So di ba, when there is injury, normally dapat merong clot formation. So dapat, pag na-serve niya na yung purpose niya to stop the bleeding, it should be removed. Fibrinolysis of the fibrinogen molecule occurs if the fibrin or the clot serve its purpose. When there is a derangement, for example, when there is excessive fibrinolysis, this could cause bleeding as a result of fibrinogen consumption as well as premature clot lysis before wound healing is established. But on the other hand, we also have inadequate fibrinolysis, which will eventually lead to clot extension and thrombosis. And that thrombosis could lead to embolus formation, thus will cause or can cause stroke. So as you can see here, the plasminogen is converted by your tissue plasminogen or TPA and urokinase to plasmin and will degrade the fibrin to form your fibrin degradation products. So also shown here is the inactivators of your plasmin. We have your alpha-2 antiplasmin, alpha-2 macroglobulin, we have plasminogen activator inhibitor 1 and 2. So hindi lang yung ating secondary hemostasis yung merong regulatory proteins. Also, we have fibrinolytic regulatory proteins. The table 35.12, which involves the proteins of the fibrinolysis pathway. We have in box red, we have the pro-fibrinolysis, we have the plasminogen, which will become activated in a form of plasmin. We have the tissue plasminogen activator or the TPA, and the urokinase plasminogen activator. So, in cases of stroke, we administer RTPA or recombinant TPA so that the clot will be dissolved right away. So ito yung binibigay natin sa mga stroke patient para mawala yung clot nila so that we could re-establish the blood flow. So in box in blue, we have the inhibitors of fibrinolysis or the regulatory proteins. We have plasminogen activator 1 or the PI1. We have alpha-2 antiplasmin and the TAFI or the thrombin activatable fibrinolysis inhibitor. So these are the proteins involved in the fibrinolysis pathway. So take note for the pro-fibrinolysis and the anti-fibrinolysis or the regulatory proteins. So, di ba, na-mention ko kanina that your fibrin, when activated or when reacted by your plasmin, will become fibrin degradation products. So, when the plasmin cleaves the fibrin or the fibrinogen, this forms fibrin fragments, namely fibrin fragments X, Y, D, E, and the D dimer or the DD fragment. So, which is composed of your two domains of D from separate fibrin, but not the fibrinogen, cross-linked by the action of 13A. So, for fragments X, Y, D, and E, They are produced by digestion of either fibrin or fibrinogen biplasmin. But uniquely for your D-dimer or your DD fragment, this is a specific product of digestion cross-linked fibrin only. So we could use D-dimer as a marker of thrombosis and fibrinolysis. For example, in cases of your disseminated intravascular coagulopathy and in other disease state, ruling out venous thromboembolism and pulmonary embolism. So when we want to measure the thrombosis and fibrinolysis, the most specific test would be D-dimer or the DD fragment. I think that's the end of the lecture for your secondary hemostasis and fibrinolysis. So take note again of these names and other names of your coagulation factors. Please remember this by heart. So this ends our lecture for your finals for Hematology MLS 417. So I would like to thank you all for your cooperation and understanding for the past few months. I hope to see you soon, our future interns and future RMTs. Thank you very much. This is your lecturer, Winwent M. Corpus. I hope you enjoyed our hematology lecture series for this semester. I hope you will pass this subject. See you soon and God bless.